Esta es la tercera y última llamada. Vamos a dar comienzo.
Tomen asiento, por favor. Continuamos con nuestra programación con la videoconferencia Microbial Colorants Production Through Biotechnology Road a cargo de la doctora Valéria de Carvalho Santos Ebinuma, de Brasil. Pedimos amablemente a las compañeras María Fernanda Cruz Rodríguez y Adamaris Mineidi Hernández Hernández del Grupo Quinto A hacer la presentación de nuestra estimada ponente.
Academic profile. Valéria C. Santos Ebinuma is assistant professor in the Department of Bioprocesses and Biotechnology, School of Pharmaceutical Science of the São Paulo State University, UNESP. She is accredited professor of the graduate program of Bioscience and Biotechnology applied to the pharmacy of the São Paulo State University. She obtained her AMC and PhD in Biochemical Pharmaceutical Technology, University of São Paulo, Brazil. During her PhD, she performed a 6-month internship at the University of Reading in England. Since 2014, Valéria Santos Ebinuma is leading the research team BioPPUL, Bioproducts Production and Purification Lab, which focus on developing sustainable biotechnological technologies for the production and purification of added value products. Currently, Valéria C. Santos Ebinuma has as her main research interests development of biotechnological process with emphasis on submerged culture and orbital shaker and bioreactor production, insulation and purification of added value biomolecules.
La doctora Valéria C. Santos Ebinuma es profesora adjunta en el Departamento de Bioprocesos y Biotecnología de la Facultad de Ciencias Farmacéuticas de la Universidad Estatal de São Paulo, UNESP. Es profesora acreditada del programa de posgrado en Biociencias y Biotecnología aplicadas a la farmacia de la UNESP. Obtuvo su maestría y doctorado en Tecnología Bioquímica Farmacéutica en la Universidad de São Paulo, Brasil. Durante su doctorado, realizó una pasantía de 6 meses en la Universidad de Reading, Inglaterra. Desde 2014, Valéria C. Santos Ebinuma lidera el equipo de investigación BioPPUL, Laboratorio de Producción y Purificación de Bioproductos, el cual se enfoca en el desarrollo de tecnologías biotecnológicas sustentables para la producción y purificación de bioproductos de valor agregado. Actualmente, la doctora Valéria C. Santos Ebinuma tiene como principales intereses de investigación el desarrollo de procesos biotecnológicos con énfasis en el cultivo sumergido con agitador orbital y bioreactor, la producción, el aislamiento y la purificación de biomoléculas de valor añadido.
Welcome, it's a pleasure having you here. The time of your presentation starts now.
Introduction. Check. I'd like to say where we are. So my university is in Brazil, in the state of São Paulo, and in the city of Araraquara. And my university, São Paulo State University, is one university in Brazil that you have multi-campi. So it is through the state of São Paulo. So we have the same university in several cities. And here we are in Araraquara. That is a very hot place, but today is very cold, but is a really nice place. Besides make a very good introduction, I have these slides that would like to explain something about my career. So I began doing biochemical engineering in São Paulo University, and after I made my master and PhD in biochemical pharmaceutical technology, and it was I had the opportunity to make a stage in University of Reading, and after I came here to become assistant professor when I created the research group called Biopool, and now I am associate professor in my university. And since the beginning, when I was in the PhD, I began to work with colorants, and so it's what I'm going to be talk today for you. So colorants are molecules used to impart color, enhance, or restore the appearance of several products. So it is introduced in food, cosmetic, and pharmaceutical, and it has to industry. If you take a round of us, we're going to see that the color is very close to us. So the colorants is the compounds that give color to these products. And we can share and call the colorants in two big groups. The first one is pigments, and which is large particle size, insoluble in the needle, and the other is dyes that is soluble in the needle. And besides the difference between pigments and dyes, they can be also separated and classified between natural identical, and synthetic. The natural ones is produced by living microorganisms, while natural identical are artificial, but they are also found in nature. And synthetic is the one that's not produced and not found in nature. They are only artificial. But when I see and we're talking about color, I said, oh, the color is very close to us. You have to think and consider about the color perception. So the foods, they have color because the capacity to reflect or emit energy, and these energies correspond to different wavelengths. When you take a look for different colors, we see that the visible light, that's what we can see with our eyes, we have a very limited range of wavelengths, that is 400 until 700. And what you see when you are looking for a food, for example, is what one thing. We have one color that is we see, and one is reflected. So we have a relation about the absorbance of color and what see. So for example, when we say that the yellow color is absorbed in 400 nanometers. However, when we see and take a look at this in spectrophotometer, for example, the wavelength is around 600. So we have a difference about the color that is absorbed by the material and the color that is reflected and we can see in our eyes. And the color perception is one thing very important because it's the literature. We can see that when you heard something that you're doing now, you're going to observe around 10%. But when you read, is about 20%, but when you see it, is around 8%. So the color perception is really important for our society and our life. So for example, we have different brands that have color that is known in the whole world. So McDonald's is one example. If you are here in Brazil, you have this color, and if you are in Mexico, you have this color, and independent of the place you go, you see yellow and red, you're going to say, oh, there is McDonald's. And the color that McDonald's is quite interesting because you have one side that's called color psychology. And in this color psychology, say that the different colors, they have different emotion in our inside of us. So, for example, when we see the McDonald's that is red and yellow, the yellow is something that's like optimism. I'm going to get it. And the red is something that is very excitement. So we have two colors that bring the sensation that good things is going to happen. And when you go there, you have this feeling. And if you see and check the different brands that we have, you are going to see that the colors and are very similar to different kinds of products. So when you consider the different foods, for example, it's quite important to see, okay, I have this, each one I'm going to show. okay, I'm going to choose the one that's not so good. For example, this strawberry, or the one that's really red and quite interesting. So it's one thing to consider, and to when you are think about the foods they are going to see. And so how the color is something so important in our society, you can check the importance of this global. So the markets of the colorant is quite important. And here I am not sharing if it's natural or artificial. The colorant market is quite important. It's growing because the color is very inside our society. However, when you consider, for example, color for food or for pharmaceutical products, we have different regulations. And so, for example, we have the FDA, that's the regulatory agency in the USA. You have EFSA, that is one used in Europe. Here in Brazil, we have Anvisa, and I have checked and see that COFEPRIS is the one in Mexico. So what these agents makes, they check and regulate the use of different additives, and the colorants is inside this classification. I take a look at and asked for the rules, a little bit about the rules in Mexico. So, for example, we have different rules. It depends on the country and depends on the place we are. So, for example, I saw that in Mexico you have acuerdo, rules that was implemented, and they have different ones for depend on the colorants going to be applied. So it's fair going to be applied in food, and they're going to be applied in pharmaceutical compounds that I said before. They have different rules. And here in Brazil, for example, we have different classes of colorants that are using artificial. And here, the artificial colorants are quite used in our products, but you have some ones that are not allowed to be used. And I checked it, and in Mexico is the same. So, for example, here we have the main colorants that are used in several kinds of products. And for example, tartrazine is very used and is used in Mexico as well. The most part of the countries use this, and they give the yellow color for different food, mainly fast foods. But I'd like to highlight this in this table, the erythrosine, because erythrosine is one kind of colorant that was permitted in Mexico, in Brazil, in European Union. However, it was banned in USA. And it was banned very recently. It was in January of 2025, and the companies have to took out it from the food and the medication as well. And as the FDA gives the rules for different countries, it's going to be studies. Here in Brazil, we continue using, and if I check what I checked, I saw that in Mexico as well, they continue being used. However, the regulation is changing every day, every time, so it can change in the future. So, and there is a lot of questions in the society about the use of food about colorants, artificial colorants in foods and in pharmaceutical products. And so natural colorants are gaining attention in the market, because if artificial colorants, they are so discussed in the society, and there are several problems associated to the use of them. So the natural colorants are gaining markets in this sense. However, when you consider the price, and we know that when you're working in different products, the price is quite important, because I'd like to know, okay, how much is going to be the cost of this my product? We see that one big problem of the natural colorants is the price. So, for example, a red, that's a red colorant very used in different compounds. It is a price around $24 to $42 the kilo. But when you see choconilla, that is one colorant extracted from insect, it costs around $50 more. And if you see a colorant produced by bacteria, for example, the price is too high. And the price is a problem. And but when you consider the difference about the natural ones and the colorants, the artificial ones, we see that they have different approach as well. So this one is one table that saw the big problems about one and the other. So, for example, the colorants, the natural colorants have soft colors and suitable nuances. To the other hand, the artificial ones, they have bright and intense colors. And this is one thing really important when we are going to apply our colorant in different matrices. For example, I saw in the beginning of my presentation the strawberries. And the strawberries that are very red, they are more attractive. And the artificial colorants can give this because they are very intense colors. And to the other hand, the natural ones, they not give so intense in colors. But we see that the preference are changing and the consumer are changing this. So today we have a more consensus about the use and the what we eat. So the consumers are looking for different compounds that are more healthy, and beside natural colorants are really good. But one aspect when you work with a process, bad for the natural colorants, the stability and consistency. So the natural colorants are not so stable when you consider pH, temperature, and light. However, the artificial ones, they are more stable over time in this process. And one thing really important is the time. Okay, it is quite stable in the pH during 5 minutes, but when you consider that this is going to be applied for during a long time, how is stable. And in this point, the artificial colorants are quite good because the stability of them and the consistency in different matrices is very high. And you have a problem about the regulation as well. So because you have to look for it and test. And so, for example, I showed for you about the erythrocin, and the erythrocin is one artificial colorant that was approved until last year, and now it changed. And we have different kinds of studies that will be performed to see if the colorants are being approved or not. And this is one thing that's quite important. So the regulation is one thing to consider when you are going to talk about different kinds of colorants as well. So what were the possibilities of natural colorants? They have two different main classes. The ones is plants, animals, and microorganisms. And when you look for these different classes, you can see that you have in the nature different plants with colors, and these colors are quite brilliant, and this is good. But you have different kinds of microorganisms and insects. The main one is the Cochonilla, as I showed before. But we have different classes, as I said, plants, microorganisms, and insects. But we have a different chemical structures as well. That's quite important. And this different structure can give colors with more intensity and can give more stable as well. So for this, it's quite important to begin to study. These are two groups that work here in our research group. One is polycetide chromophore, and the other is carotenoids. That's also found in different plants. And is the main compounds that you work here. So in nature, we have several microorganisms that are able of producing these colorants. So we have Serratia that produce prodigiosin, Chlorella that produce different kinds of green compounds that makes photosynthesis. You have Rhodotorula and you have Thalaromyces, that is one group that you work here as well. But okay, these microorganisms produce colorant, but why they do this? So the first thing, they are obtained from partially colored compounds and they works and play important roles in the physiology and the molecular process of these microorganisms. Second, it presents several functions in the body of the microorganism. So they are compounds that are really important to the metabolism of the microorganism. And third, they work as defensive systems. So for this reason, a lot of media that the colorants are presented, they also present biological activity. So it's quite common. We take a look of extract with a colorant, and there we have also biological antimicrobial and antioxidant activity as well. So in this sense, as I said, these colorants or these extracts containing the colorants, they can protect against UV rays, they act as antioxidant products, protect from extreme conditions of cold and heat, and they have antimicrobial activity as well, and they are used as acquisition of nutrients. So, for example, iron, nitrogen, and carbon. So the production of colorants are quite linked to the metabolism of the microorganism. And the production of colorants is interesting from an industrial perspective. So we know that the colorants are produced because of the metabolism of the microorganism, but it's interesting for the industry. Yes, it is. And it is important because the microorganism can have growth rates, high yield, and we have, as I showed before, a variety of colorants produced. We can say that this colorant is going to be produced in conditions that to be controlled. For example, temperature, and we can minimize the variations batch to batch between the production steps, and the colorant production are more stable than the one produced by plants or animals. As I showed before, they are not more stable than artificial, but when you compare within the class of natural colorants, they are more stable. And they not present problems of season, so we can produce colorants for the whole of the year. It is quite important. And when you work and establish the bioprocess, it is really fast and the yield can be really high. So the production of microbial colorants has two approaches. So first is finding new colorant producing strands from the diverse of high environments we have. And second, achieve high yields in order to have these colorants able to be applied in different kinds of industry. Because if you consider one thing is that, okay, you have color in all the place. But if I produce a colorant by a microorganism, I have to secure that I have a colorant in the amount necessary for the company or for the process. So in this sense, change and list has to be overcome. And between these is that works with conditions to improve the stability of these colorants, works with different kinds of materials that has equivalent in terms of color between natural and artificial ones. We have to got the regulatory agency approval and development of these colorants in temperature, light, and acid conditions, and scale-up state. And really important when you consider for the industry, it's about the cost, because the artificial ones has the price really low comparing with the natural ones. So, which is the success of a microbial colorant? Reduction in production cost, regulatory agency approval, and market acceptability. In this sense, the bioprocess development is quite important. And which is a bioprocess? So bioprocess is a specific process that uses completely living cells or their components to generate value-added products. So we have a different microorganism. We give for this microorganism a raw material, and the microorganism is going to metabolize this raw material and it's going to produce products. And we have different kinds of products produced by the microorganism metabolism. So antioxidant products, fermented foods, enzymes, colorants, organic acids, insecticides, antibiotics, between others. So consider the bioprocess. If you are working with aerobic microorganism, we are going to give to them oxygen, a nitrogen source, carbon and energy source, mineral components, so different kinds of salts. And after the metabolism of the microorganism, they are going to produce CO2, biomass, and the biocompounds of our interesting. But the way that the microorganism is going to metabolize these different compounds is quite important, and it is studied in the bioprocess development. So if you consider the phase 1, we're going to have the microorganism selection. After, we have to make a characterization of the microorganism and the bioproduct. It's quite important to know about the characteristic of this microorganism, of this bioproduct. So, for example, about the stability of it and the chemical structure, that's quite linked with the chemical structure of it, but how it works. So, make small scale, that is works one in shakers, and after make a scale up of the process. And when you consider it, we can have different kind of bioreactors. And for the last phase is the extraction and or purification process. And here I'd like to highlight the phase 2 and phase 4, because when you works and says about small scale experiments, we can work with shakers. That is one about the first figure here. But we can also make this in bioreactor. But the bioreactor, we can have bioreactor in lab scale, that is until 10 liters, and we can have bioreactors for industrial scale, that is from 50 liters, for example. And this is one point quite important, because we can direct and make different in the production process, working with different aeration, agitation speeds in order to promote the production of our compounds of interest. Considering with the points and development of the production of bioproducts, we can say that the first step is going to be the selection of the microorganism, and characterize the microorganism and know about which species it is quite important, because you can direct and after make a relation with the metabolomics works, for example. Make a color characterization and identification of the colorant agents, and have the knowledge about the functionalization and working, okay, I have this colorant, when it's going to be applied in the food, the pharmaceutical industry, and after make this optimization of the process. And the optimization of the process has different steps. So the first one is going to make the selection and the optimization of the culture media, and after making the selection in bioreactors to make this relation about the aeration and the agitation of the process, and in order to have the amount necessary for the products of your interest. So consider it, we have different biomolecules that are working, and they have some ones that is research steps and the other is industrial step. Here is some examples that is in research step. One of them, the polycetide red, here elucidated, is one example is Saccharomyces amestocii. That's a microorganism that work here in our research group, and it produce colorants that's the color range from yellow until red, working there with the orange one. And this polycetide groups is the one that I showed in the first slides, but you have different ones. So for example, melanin, it's a black colorant, naphthocnone, cataxanthin, astaxanthin, but these colorants are in research step. They are only in the lab. They don't have this in the market. And I'd like to bring this other table that shows some colorants that are used in industrial step. So one good example is our pinky red that is anthracnone group, and it's producing by penicillin oxalate. And this is one example of colorants that are used in different kinds of industry, mainly in the food ones. But you have also riboflavin and beta-carotene. Beta-carotene can be also produced by Bloschella trispora and Dolonella selina, but these two microorganisms are used in industrial step. So we have different ranges, and it's quite interesting when you consider, okay, we have one colorant that is in research place, and now it is industrial scale, which is the steps from it. And but we are giving and getting different market, and the color stability is one thing that's quite studied, but you have now different approach that can improve its characteristic. I'd like to know about, I like to tell you about one kind of colorants that are very used mainly in Asia countries, and the one is monoscous colorants. And the use of monoscous for the production of this polysaccharides that's classified as azaphylline colorants is very old. In Asia countries, they have a fermented rice that use monoscous as micronutrients. And so we have different now azaphyllones that are called monoscous colorants because of the chemical structure of these compounds that are quite similar to the monoscous ones. And they are divided it in United States and the European Union. They have not approved monoscous colorants because of one specific compound that is called monocholine. However, there are a lot of studies changing the culture media, changing the microorganism, and although it is not approved, they have a lot of studies make that this compound is not going to be more produced in the culture media. And it's quite effective and is approved to use in Asia countries, for example. And we have one product that is called Anscacin, that is approved by FDA and contains two active ingredients, monacin and acamflavin, but not contain monocholine, okay, because it was moved about the purification process. And I'd like to tell you something about our works and the biopool objectives. So in our research group, you work with microbial colorants, and our main objective is make the bioprocess development. So one of the microorganisms that you work is Rhodotorula glutenis, Talaromyces amistokia, Streptomyces lavanduli, and Fusarium explora. So we have Rhodotorula that produce carotenoids, and I am going to tell you something about our research here. And Talaromyces that produce red colorants, very similar in terms of chemical structure of the monoscous colorants. Streptomyces lavanduli that produce a blue colorant, and Fusarium that produce a colorant very interesting because the color change according to the pH that we are working. I'm going to tell you today about two works of a research group, but they are works that are a continuation of one of others. So the first one is the beginning of our work here with Rhodotorula glutenis, and we have different doubts about the production of these carotenoids, and it was work developed in collaboration with Professor Kasamo Musagi and Professor Jorge. And in this work, it was the first work that we made here in our lab with this strand. And we know that to a yeast produce carotenoids, we have to consider the nutritional contents, temperature, fermentation time, nitrogen source, carbon source, and pH. And the first thing is that we thought is how the effect of inorganic and organic nitrogen sources on the production of carotenoids by the yeast. And so it was made different approaches using statistical optimization designs to improve the production of the carotenoids. And this was what we did. So we have this microorganism in glycerol medium, and you make this propagation of the microorganism, and you use statistical tools to improve the production. So we made several experimental designs and used the surface methodology to improve the production. After we have developed the nutritional media, we made different experiments in bioreactor, followed by the separation. In this case, the carotenoids produced by VCS, it is intracellular one, which means that the microorganism did not export the carotenoid, it gets inside of the cell, and you have to make extraction to get this carotenoid. So this was the approach used. Take the microorganism, make experimental designs, changing different nitrogen source, and this is quite important because in this work we used asparagine as nitrogen source, and we began trying to take this from the culture media because mainly of the price. And in the beginning, you had other objective, the use of asparagine, but it was improvement of the culture media. And when you make the experiments, we know that these microorganisms are able to produce 3 carotenoids. So the beta-carotene, the toluene, and the toluene. And at this point, it was important the reference and the knowledge about the microorganism and the compounds that you are working. Because making a research in the literature, we saw that toluene, it was compound that have antioxidant power very high, and compared with beta-carotene, it was more pronounced, this characteristic. So it was one thing that we got attention and we like to work more. So we have these 3 carotenoids, so we began to work. In the first experimental approach, you analyze it: glucose, phosphate, sulfate, nitrate, and the pH influence in the production of the whole carotenoids at 30 degrees, 170 RPM for 72 hours. And we saw about the relation of the production of cells and the lipid content, because the yeast are able to produce lipid as well. And we have this production of the carotenoids used. When you analyze it through the using the statistical tool, we saw which was the main parameters, the main variables for this process, and in sequence we saw and analyzed that they make more influence in the production of beta-carotene and toluene. Mainly was the glucose, the interaction between glucose and nitrate ammonium, phosphate, and nitrate ammonium. So, and toluene was mainly effective by the interaction between glucose and nitrate ammonium, phosphate, and pH. And this is different because when you consider the metabolic route for the production of these different carotenoids, we see that the toluene is one product produced, and after and with this oxygenation, it is produced toluene. So we made other design, now working with glucose, phosphate, sulfate, asparagine, and pH. And now we see that we changed the nitrate for asparagine, and the production of the carotenoids were improved as the production of biomass and the lipid content. And with this approach, we improved the production of the whole carotenoids in our media. And the Pareto chart showed us which is the influence of the different variables in our process. So it's important to see that worked with inorganic and organic source, and both influencing the process, but it was not they're not the only products that influenced in the process. And so for this, it's quite important to analyze which we wanted. So the statistical tool is one tool, that's statistical design is one tool that can be used to improve the process, but you have to pay attention in all the results achieved and the relation and how it's related with the microorganisms that they're using. So after we made used the response surface to optimize the production of the carotenoids in this point. And we saw that the pH and nitrogen combination influenced the process of the production of the colorants, and the combination of asparagine and inorganic source also favors the production of the carotenoids. So after we made a production in a bioreactor, and we see that the production of the whole carotenoids improved. So we have here the production of the toluladine and toluene very improved, and low production of toluene. It was quite interesting as well, because our main products that were interesting was beta-carotene and toluladine. But we could analyze the whole process. So, for example, see when the glucose was consumed in the whole process, and how it was the consumed processes in the synthetic process. So we saw that it was the significant importance and the improvement of aeration and agitation can improve the production of the 3 carotenoids in this point. The main conclusion of this work is that the metabolic rate is quite important in order to produce the different carotenoids. So we have first produced toluene, beta-carotene, and after toluene, and after toluladine, but we improved the production. However, we had some challenges and open questions remained in this project. So substitution of asparagine in the culture media, and favoring toluladine production over toluene. So we consider, okay, we wanted to took out the asparagine from the culture media, and as toluladine has a very high antioxidant activity, it is important to our media, and we'd like to improve its production. In this sense, we have to make more work and more research in order to get these answers. And this is the work of other students here in our research group, that is Julio, and he made the process very similar in the beginning. So they had got the microorganism of the torula, made the cultivation, and made the extraction. But now we changed the extraction process, because now we used bone meal and acetone as solvent to make this extraction. We got the 3 carotenoid as well that had this really nice color, but we'd like to improve. And as I said before, we had problems. So we made and used statistical tools to improve the production. And here I show that the standard condition that was using asparagine, and the result of the first of the last experiments I showed for you, you had 0.49 milligrams of ml of toluladine. And so we made the first experimental design, improving the production and working with different variables of the process. Works also with different nitrogen source. We tried to improve and use surfactants, because we consider that if you had surfactant in the media, the cell wall is going to be easy to make the cell disruption. And when you have added the surfactant addition, you use Opter. We made other statistical design in order to optimize the studies. We made the studies with light, which you see that did not have influence in the process. And after we made kinetic studies, improved the process. So all this approach used was in order to change the culture media, and changing the culture media, improving the production of our target molecule, that in this case was toluladine. Comparing the first condition and the last one, we have a considerable increase in the production of the carotenoids. And this is one part of the work that we have. It was also published. And we have thinking, and as it began, where we can apply this colorant. And one answer and one thing that we tried is to make the application of the colorant in gelatin. And it was very interesting results because we had the gelatin with color. And when we make the stability of the gelatin and the color of the gelatin, it keep it for a long time. We made analysis for 24 days, and the result was quite similar. We didn't have a lot of loss of the color during this. So my final considerations is the forecast for the use of natural colorants is promising for the future because you have market for it and you have different microorganisms in nature that can be used, and we have a very great chemical diversity of microorganisms and colorants to be explored, and the bioprocess is quite close to this to make this available for the market. The great obstacles for exploring new colorants are the current legislation, because as I showed for you, you have a very regulation that asks for toxicological tests depending on the application. The cost of the colorants produced by microorganisms, because they are more expensive than the one produced by chemical routes. However, if you work more with the bioprocess, as I showed for you, you can improve the concentration of the colorant and also reduce the cost and the consumer acceptance. We have to go to our consumer and say, okay, we have these colorants that are not toxic and they can give color that's is quite good for your product, and okay, you can use it. But you have fair markets, and when you consider that biotechnology is increasing and the tools and technology is increasing very, very fast, you can see that this market is going to be very full of natural colorants in the future. I would like to thank you, my research group that's called Biopool, and I invite you to follow us in the Instagram. So I have here in this picture a lot of students that works in this process, in this project associated with the production of natural colorants. And I would like also to thank you, the organization of this week, for the invitation for talk. It was really good for me share this knowledge that you are working here, and invite you to follow our research group, and invite as well to come here to Brazil and make research with us. And so, gracias, and questions, I am open to answer it.